Today we'll be discussing peptic ulcers.
Well, Peter, can you stop the recording?
Peptic ulcers are painful sores that develop in the epithelial lining of the stomach. These peptic ulcers can cause individuals to develop dull upper abdominal pain, nausea, vomiting, heartburn, and fatigue. If left untreated, peptic ulcers can lead to internal bleeding, and excessive bleeding can be life-threatening. But what causes ulcers?
Back in the 80s, Giulio Bizzarra proposed that the stomach was sterile. Other researchers thought ulcers had a genetic basis, were linked to cancer, and were caused by having too much stomach acid or by certain lifestyle factors. Antacids, surgery, and lifestyle changes were recommended as the only treatment for peptic ulcers. Researchers Barry Marshall and John Lykoudis, however, knew that antibiotics could cure peptic ulcers and that a bacteria called Helicobacter pylori, or H. pylori, was associated with 100% of the ulcers. However, there was a lot of doubt and pushback from the scientific community. You see, Marshall's ideas failed two of Koch's postulates, number 1 and number 3, but Marshall was determined to prove his theory and took matters into his own hands. He drank cultures of H. pylori from the gut of a sick patient, and after a period of vomiting, diarrhea, fatigue, and pain, Marshall developed ulcers and biopsied his own gut. He found culturable H. pylori and then used antibiotics to treat himself, supporting his theory. Because of this experiment, we now know the true causes of peptic ulcers and the mechanisms of H. pylori.
H. pylori is a gram-negative pathogen that swims out of the stomach lumen towards the mucosal layer. It chemotaxis to areas of higher pH and corkscrews into the mucosal layer. Then, H. pylori produces urease enzymes and secretes ammonia to buffer the low pH of the stomach, which allows the local region around H. pylori to be in higher pH. Urease and other enzymes break up the mucosal layer and extracellular matrix. Many H. pylori gather in the same area and create a high local concentration, which allows them to cross the diffusion barrier created by the mucosal layer. H. pylori then uses type 4 pilus and HPAA to attach to the cell surface of the epithelial layer. The bacteria injects toxins like Kag-A, which causes cell damage and changes cytoskeleton structure. Once crossing the epithelial layer, H. pylori uses Vac-A to create intracellular vacuoles inside the host where it can live. Vac-A eventually leads to mitochondrial dysfunction, apoptosis, which ultimately creates stomach ulcers. However, only 10 to 20% of H. pylori infections actually lead to an ulcer. In fact, current research is looking into the benefits of having H. pylori in our stomach. Recent studies by Zahiri even found potential protective effects for allergic asthma by regulating ratios of helper and regulatory T cells. The researchers have even suggested applications of H. pylori in creating therapeutic products to prevent or treat asthma. However, this research is still in the works, so don't go around drinking H. pylori.